अंकिता मैम एडमिट कर रहा हूँ सबको.
How are you? Nice to see you all.
Hi, sir. Good morning. Good to see you.
Good morning.
Morning.
Hey,
Morning.
how are you?
Fine.
Good. Nice to see you all.
Yeah.
Hello.
Hello.
Yes, sir. Good morning, sir.
अच्छा, ठीक है ठीक है. Hello, hello.
So we wait for 5 minutes.
Yeah, I'm ready when you are.
People are joining.
Absolutely. No, 5 minutes is, I think that's the time, right? So 5 minutes is great.
Yeah.
Yeah, no problem.
Hope you are not feeling sleepy.
Not at all. Not at all. This is, I'm not in bed yet these days.
Yeah.
और सर नमस्कार.
Namaskar. Keep logging from another account. फिर आना है बाद में. But आप इंट्रोड्यूस कर. आपका नाम क्या? For a victory, it's early in India, so people are joining.
That's fine. Yeah, I understand. I understand. It's very early.
Yeah, but then we also didn't want to delay it too much. Would have been very inconvenient for you.
That's fine. Thank you. I'm ready. It'll be fine. I appreciate that.
Manuals.
Are people going to work these days, or everybody staying at home still?
Yeah, it's almost open now in India.
Nice.
Yeah, so it's even schools and colleges, universities are gradually opening up now.
That's excellent.
Yeah,
It's good to hear.
but it has not started yet in US, right?
It's still pretty, some places are more than others, but we're still pretty much locked down, although the labs are open. So the people are working in labs, but the classes are all online.
But in Delhi, actually, the cases are substantially come down now. I don't know why, but we have very little positive rate now in the country now. Gradually coming down.
Yeah, that's great.
Let's hope it gets better.
Yeah.
Because become very difficult for us to do everything online, and we are not really enjoying doing things online.
Yeah, it's no fun. It gets tiring. It's very tiring. Have they been doing a lot of vaccination there?
Yes, they are doing a lot of vaccination now,
Good.
and things are moving in the right direction. That's what we are hoping.
Mm-hmm.
Yeah. Yes, shall we start now?
Can we start, sir?
Yeah, we start. Yeah,
Absolutely.
Ankita, go ahead.
Thank you.
Absolutely.
A very good morning, everyone. We welcome you to the third and the final day of the 61st Annual International Conference of Association of Microbiologists of India, AMI, and the fifth annual conference of Indian Network of Soil Contamination Research, INSCR, in association with the University of Delhi, Indian Agricultural Research Institute, the Energy and Resources Institute, TERI, and Indian National Science Academy, INSA. The last 2 days of the conference have been an ocean full of knowledge, and moving towards the last leg of this conference today, we will learn about the latest research in the field of microbial systematics, translating innovative ideas into enterprise and microbial pathogenesis. Without taking any more of time, let me introduce you to our first plenary speaker for the day, Professor Victor D. Rita, President, American Society for Microbiology. I would request Professor Ruplal to formally welcome Professor D. Rita.
Professor Victor Rita, thank you very much for being with us for the second time. I know it's late hours in the US, but you still made it for the conference. In fact, many of my fellow colleagues have been asking me, how did you get such great scientists to the conference? So it's also a surprise for me that you have so much passion for science, and even Edward DeLong, you, Cantemis, everybody was there. So thank you very much. And you all know Professor Victor De Rita is the president, American Society for Microbiology, chair for ASM membership, and he's the editor Bacteriology Journal. So thank you very much being here. I request now Ankita take over and then give the floor to Professor Victor De Rita. We are excited and very eager to hear his great science which he is doing. Thank you very much.
Thank you, sir. Professor Victor De Rita is Rudolph Hu Endowed Chair in Microbial Pathogenesis and Chairman of the Department of Microbiology and Molecular Genetics at Michigan State University. His work focuses on molecular mechanisms of bacterial pathogens, with particular interest in gut pathogens, Vibrio cholerae and Campylobacter jejuni. He also serves as chair of the ASM membership board and editor for the Journal of Bacteriology. I request Dr. Rajendra Prasad from INSCR, Dr. V.C. Kalia, Konkuk University, Korea, to chair the session, and Dr. Shelly Anand from Deen Dayal Upadhyay College, University of Delhi, and Dr. Jaswinder Kaur, Gargi College, University of Delhi, to co-chair the session. I request Professor Victor to deliver his lecture on the topic entitled Growth Physiology of Enteric Pathogens During Infection. Over to you, Dr. Victor.
Thank you very much, Dr. Dua. I appreciate your very kind introduction. And Roop, my friend, always so nice to see you. And VC, very nice to see you. So thank you for the invitation. It's just it's a real honor to be able to be speaking to you, and I wish I could be there in person. I was there before in 2013, had a terrific time meeting so many great people and great scientists, and I had a fun time the other night talking about scientific communication with Roop and others, and I actually learned, I told you, Roop, I learned a lot. I was very inspired by your wisdom on that. So I want to talk today about our science, not so much about just science communication. I hope I communicate it well, but I want to talk about the work that we've been doing, in particular on Campylobacter jejuni. As Dr. Dua said, we work on a couple of different pathogens, and because we only have about a half hour, I'm going to talk about one bug and one bug only, and that's Campylobacter jejuni. The general topic and the general title that I gave on understanding metabolic questions and metabolic pathways in enteric pathogens is something that we've become very interested in the last several years. And I'm going to talk today about the role of lactate and lactate metabolism in the pathogenicity of Campylobacter jejuni. And we use this model, the ferret model, to ask the questions that we're asking. And I'll explain all about that as I go through. I want to give all the credit to this work to Ritam Sinha, who is actually here today. He was able to make it to this talk. Very proud to show his work. All of the work I'm going to talk about is from Ritam. Dr. Sinha did his PhD at NICED. He's a native from Calcutta, terrific scientist, and has a bright future in front of him. And this is all of his work. We collaborate with Maddie Keipel, who's a veterinary pathologist, and Nesh Stopnisek, who's a bacterial bioinformatician that's helped us a lot with some of our microbiota work. I'm not going to talk anything about work we've done with Jan Dufour and his student Nguyen Nguyen on motility in Campylobacter, but I'll be talking about the work of these people. And again, this is all led by Dr. Sinha in the laboratory. Okay, so we're studying Campylobacter jejuni, and a little bit of background on Campylobacter. This is one of the most common bacterial causes of human gastroenteritis in the world. We think of Salmonella, we think of Shigella, certainly we think of Vibrio cholera, not a gastroenteritis pathogen, but a diarrheal pathogen. But Campylobacter kind of comes in under the radar sometimes. We don't think about it quite as much, but it's a highly prevalent microbe, and it lives most of its time in the gastrointestinal tract of poultry, of chickens and turkeys and ducks. And that's where we encounter it because we eat the meat from these animals, and that meat can be contaminated during preparation of the meat after slaughter with the gut contents, and those gut contents can then be consumed by humans who, when they consume it, have a completely different intestinal experience with Campylobacter than chickens do. So whereas in chickens, this is a commensal microbe and it colonizes to very high levels without any symptoms in the birds, in humans, we get this massive inflammation, lots of diarrhea, some blood in the stool, lots of neutrophils. So it's a completely different outcome in humans than it is in poultry. And we've done a lot of work in poultry to understand it. In fact, Ritam had a very nice paper last year understanding some of the metabolic functions of Campylobacter while growing in poultry. But now we're interested in understanding some of the disease mechanisms in the human. And so for that, we need different models. So the unresolved questions that we have that we're going to look a little bit about today relate to very simple things, but things we don't know the answer to. What are the carbon sources this microbe uses? How does it grow? How does it thrive in the intestine during infection? What role, if any, does the microbiota play in the colonization of Campylobacter? And then what role and what are the source of gut metabolites that influence the pathogenicity? And I'm going to talk a lot about the metabolites, and I'll talk a lot about the carbon sources. We're not going to talk so much about the microbiota, although the microbiota are always present in both our minds and in the experiments because they play a big role in the gut, obviously, in the healthy gut. So largely the way we think about this is Campylobacter comes in early on in very low numbers. It colonizes in the intestine, and then something occurs in the intestine driven by the microbe undoubtedly, but obviously in cooperation with the host to enable this population to expand quite extensively. Inflammation occurs, and this is the disease pathogenesis. Lots of bacteria, lots of inflammation leading to damage to the intestine and diarrhea. The issue with this microbe, I'm getting a little bit, yeah. The major issue with this microbe is we don't have a great animal model to study the disease traits. We can study the commensal traits in chickens. We've done work with the chicken quite a bit in my lab, but for looking at disease, people have used immunodeficient animals, animals treated with antibiotics to destroy the microbiota or to create a dysbiosis. But a simple model where we can simply infect naturally animals has been lacking. So this is one of the things that Ritam came to my lab to be able to establish. And what he did was establish a ferret model. And I'll talk a little bit about that and what that model looks like and what some of the things we've discovered. But I want to give you essentially the punchline, the answer, the things that we're thinking we're discovering and that we're exploring further. So we think that early on in infection, the microbes are colonizing essentially a healthy gut. A healthy gut has low oxygen levels in the lumen. This microbe has to acquire oxygen in order to grow. And we think that the inflammation leads to alteration in oxidative metabolism that ultimately results in lactate production in the GI tract. And that lactate can cause growth of Campylobacter. It's oxygen dependent, and we'll talk about that. And we also have evidence that lactate induces motility of the bacteria. I'm not going to show you any data for that, but Tom has done some very nice single cell imaging work with Jan Defore's lab to show that lactate can stimulate motility of Campylobacter, and we think that motility plays a major role in the ability of this microbe to thrive as well. So this is essentially what we think is going on, and now I'm going to talk about the experiments that we've done to suggest that. When we start this work, we think of models of the intestine that we and others have explored. But when we think about the intestine, we have to think about the healthy colon first. All right, the healthy colon has a normal microbiota in this luminal space. These are the epithelial cells. This is the crypt, and the crypts are cells that are undergoing replication because the crypt colonic cells are migrating up to become part of this barrier function, the epithelial cells at the top of the villus tips. So the crypt is probably very rich in oxygen because the metabolism of the crypt cell is different than the metabolism of the barrier cells in the epithelial surface. The barrier cells, stimulated by butyrate produced by the microbiota, are undergoing oxidative metabolism, so they're consuming oxygen. So this is an anaerobic environment or a lower oxygen environment, and the crypt tissue is a much more oxygenated environment. In fact, we've done a lot of work with Vibrio cholera, which colonizes crypts very well, and we've shown that Vibrio uses oxidative metabolism to grow in the crypts because there's a lot of oxygen there, and they can survive without their anaerobic respiration. But with Campylobacter, they don't go into the crypts, so they have to live in this area. And so when cells become damaged by infection, and this is a model of Citrobacter infection that Andreas Baumler's lab and many others have worked on, in the presence of some microbes, inflammation occurs. Neutrophils can be pulled into this environment because of the infection, and the neutrophils cause damage to the epithelial surface of the host. They also disrupt the microbiota, and that alters the oxidative metabolism of the epithelial surface, and the damage that occurs causes hyperplasia, and that hyperplasia needs to be repaired. And so there's a lot of cell repair and proliferation in an inflamed intestine. Oxygen metabolism is altered because butyrate is no longer being taken up by these colonic cells that are repairing this hyperplasic lesion. And so oxygen levels increase, butyrate levels are higher, and we think this is an environment where some pathogens can thrive. And we think that that's essentially the environment that Campylobacter is either inducing or benefiting from during infection. So the outline of the talk is to talk about the ferret model, to show you what Tom's done to develop this model very nicely in the lab, and then to look at lactate metabolism, because as he developed this model, he discovered that lactate concentrations are elevated in the inflamed intestine, and we think that that's driving growth of Campylobacter. So those are the two aspects of this infection that I want to talk about, and we'll talk about a few other things along the way. So the ferret model is a nice one for us for several reasons. First, we can colonize these animals without first treating them with antibiotics. We don't have to disrupt the normal microbiota for Campylobacter to colonize. A lot of mouse models that people use are dysbiotic mice models because they provide antibiotics before infection, so the microbiota is disrupted. We can inoculate through the oral route, which is the natural route of infection in humans. And these animals, most importantly, are immunocompetent. So there are a lot of mouse models that people have developed in addition to antibiotic treatment to disrupt the microbiota. There are genetic lesions in the immune system to make the mice more inflammatory or otherwise disrupt immune cell signaling. So the nice thing about the ferret model is we don't have to do any of these things. It doesn't mean we don't have to do some things in order to get the ferrets to be infected, and I'll talk about that when I describe the model right now. So this is our basic timeline of the model. We take these animals out for 3 days after infection. 2 hours before infection, we withdraw food, administer Zantac, which is an over-the-counter essentially neutralizes stomach pH. So it's something for heartburn, essentially heartburn medication that people use. So we essentially neutralize the gastric pH because Campylobacter is sensitive to pH. We infect at 1 hour, we infect at time 0, rather 2 hours after treatment with Zantac. With 10 to the 9th CFU of Campylobacter, we use this strain, 11168, but from now on, I'm just going to say Campylobacter jejuni. We infect in a bolus of sodium bicarbonate, which also helps to buffer the inoculation to keep the Campylobacters happy as they go through the stomach. An hour and 5 hours after we gavage them with this bolus of Campylobacter, we give Lamodal. Lamodal is an over-the-counter drug that inhibits peristalsis of the intestine, essentially represses the diarrheal response and the intestinal movement response, so the microbes stay in the intestine long enough to begin colonizing. And that's just these are the 2 steps we have to do to get this model to function. But no antibiotic treatment in these animals are immunocompetent. 24 hours and 72 hours, we collect a lot of tissue, stool, intestines, organs, and we measure CAMPy. We do immunohistochemistry to look for CAMPy, look at markers of inflammation, damage, and damage repair mechanisms that are all hallmarks of inflammation, and I'll talk about those as we go through. All right, so let's first look at the Campylobacter loads in the stool. So this is what comes through the animal after 8 up to 72 hours after infection. All of these experiments are done with several animals, and several animals were also infected with or gavaged with PBS, and we don't ever pull Campylobacter out of the PBS infected animals. So these animals are not naturally infected with Campylobacter when they're in our vivarium unless we give them Campylobacter. So the blue marks are the infected animals and the red marks are PBS, but there's no recoverable counts. So at 8 hours, we see the stool has a fairly high amount of Campylobacter in it. 24 hours later, the amount in the stool has diminished. So there's a loss of colonization for the first day of infection. And then over time, the colonization loads as measured by Campylobacter in the stool come back to very high levels, so that by the end of the experiment, we're getting loads of Campylobacter up to 10 to the 10th grams of 10 to the 10th colony forming units per gram of tissue. So high levels of colonization. Let's look at this a little bit more carefully now, looking at at day 1 versus day 3 and different tissue types. So day 1, we see within the first day, the colon. So now we're looking at down deep in the intestinal tract. The colon has about 10 to the 5th CFU per gram of tissue. The small intestine is colonized, but to pretty low levels, about 100 microbes per gram of tissue. The mesenteric lymph nodes, which drain this site, are not really affected yet, and the liver and spleen are sterile. By 3 days, the colonic levels are very high, so this is starting to reflect what we see in the stool, up to 10 to the 9th, 5 times 10 to the 8th, 10 to the 7th, up to 10 to the 9th CFU per gram of tissue. The small intestine is colonized pretty heavily too, although a couple orders of magnitude below the colon. The mesenteric lymph nodes, liver, and spleen are starting to provide evidence of Campylobacter being cleared out of this infection and ending up in the reticuloendothelial system where we would expect them to be. Pay attention to these numbers. On day 1, the colon is infected, but not to particularly high levels. But by day 3, the colon is infected to very high levels. And these animals are in distress. They have diarrhea. They're sick. They look like they have gastroenteritis caused by Campylobacter jejuni. So it's a great model. And I'll show you some of the histopathology that we see. So we see on day 1, we see mild inflammation. As I showed you, we don't have that many bacteria, about 10 to the 5th CFU per gram in the colon at day 1. This is a PBS infected animal. This is a map of the normal mucosal epithelia with the villus cells in the crypts that I showed you earlier, the submucosa and the muscular layer and the lamina propria underneath the villus epithelium. So this is a PBS infected animal stained with H&E just to get some structures. This is a normal looking intestine by all measures that our pathologists can tell us. The Campylobacter infected animals, we don't see that much disruption to the epithelium yet, but what we can see in this, and it's hard to see maybe on the slide, but the little dots that we're seeing, those are neutrophils. So we're starting to get leukocytes pulled into this site because Campylobacter is present, the immune system is on guard, and leukocytes are coming in. If we look for Campylobacter by staining with anti-Campylobacter antibodies in this tissue, the PBS infected tissue, we don't see any Campylobacter, but we start to see Campylobacter on the villus surface, still in the lumen to some extent, but beginning to be covering on the villus surface. These little brown specks are the Campylobacter. Later on, we're going to look at some more damage in this tissue, and that includes evidence of tissue repair and hyperplasia. So at day 3, we're starting to see much more elongated crypt epithelia. The colonic tissue is beginning to be more damaged and more evidence of damage, and I'll show you some more markers in just a second. But now we're starting to see a thick lawn, essentially, a thick carpet of Campylobacter on the surface of this epithelium. The Campylobacter here is stained in brown on this immunohistochemical stain that we use. So the PBS animals have no Campylobacter in them, and the Campylobacter infected animals at day 3 are starting to look like they're pretty luxurious with Campylobacter. All right, now remember what's going on in this tissue, we think, is that in the inflamed Campylobacter colonized tissue, there's a lot of rearrangement of the epithelial surface so that the crypt colonic tissue is now in hyperplasia, and we're getting the crypt tissue essentially migrating and lots more cell proliferation because of the colonocytes. And we also expect to see some metabolic changes that I'll show you in just a second. So as looking for a marker of cell proliferation, we use a marker called Ki-67, which is used in cancer biology quite a bit to look at proliferation and repair. This stains cells that are undergoing replication. So most epithelial cells are not undergoing replication, and PBS infected animals are normal animals. The only replication you see is in the colonic enterocytes deep in the crypts, where we know colonocytes are replicating because they replicate to move up and become differentiated. But when we infect with Campylobacter by day 3, we're starting to see a lot more cellular proliferation, and this is a marker of cell repair because there's been damage of the cells of the tissue, and the colonocytes are now much more elongated, and the hyperplasia is being sort of the marker, the Ki-67 marker is telling us that there's hyperproliferation and cellular repair. Cells are replicated in this tissue, not just in the crypts. If we look at goblet cells, which produce mucin, there's a decreased amount of goblet cell production, decreased amount of mucin. So this is a PBS treated animal stained with a stain called Alshin blue, which stains mucin. You can see these goblet cells with lots of mucus inside of them. And when we infect with Campylobacter, we see that the mucin staining the goblet cells that produce mucus are much more diminished. Similarly, if we look at smooth muscle actin, which is a marker of inflammation, myofibroblasts are repairing tissue. This is essentially a wound site. There's a lot more smooth muscle actin staining in Campylobacter infected tissue than there is in PBS infected tissue, and it's disorganized, suggesting that essentially it's wound repair in the intestine. So that we've seen all the markers of inflammation. We also looked at true inflammatory molecules, IL-8, IL-6, TNF-alpha. These are cytokines that are produced in response to infection. They're pro-inflammatory. They help call in neutrophils. And there's a several-fold increase of these cytokines in Campylobacter infected tissue. INOS is up, another marker of inflammation. HIF-1 alpha, which I won't say anything about, but plays a role in oxidative metabolism and repair. And then IL-10, which is an anti-inflammatory cytokine, is down, suggesting that, in fact, there's inflammation going on. In fact, the mouse models that people have used for Campylobacter are IL-10 knockouts because when you lose this arm of the immune system, you become hyperinflammatory. And so that's a good model when you want to get inflammation in the gut, is to wipe out the anti-inflammatory. So in these naturally infected animals, IL-10 is diminished, which tells us likely that there's a pro-inflammatory outcome, which is exactly what we see. So everything's looking like a good infected tissue. We look at the microbiota using 16S sequencing. The ferret infected animals, the pink are the control, PBS infected, and the green are the Campylobacter infected ferrets. This is a principal components analysis of Campylobacter infected ferrets and Campylobacter infected chickens. What we see is that there's no distinction in the populations of ferrets and the microbiota populations. What we're measuring here is something called the Shannon diversity, which is just the diversity, the overall diversity of the microbiota. The ferrets that are infected and the ferrets that are not infected essentially overlap in their microbiota profiles. So there's really no difference in the microbiota in the ferret in the ferrets infected with Campy. Unlike in chickens, which we've published already, when Campylobacter infects, there is a distinct microbiota in those birds compared to the microbiota in uninfected animals. So this tells us that at least in day 3 of infection, we're not seeing any, the infected animals have no different overall microbiota than the uninfected animals. We do see, if we look at specific species in the microbiota, there's a lot of Campylobacter, which is the red bar. That's not surprising. These animals are infected with Campylobacter. And we see an increase in the infected animals, which are on the right side of this graph, of Clostridium. Clostridium is a butyrate producing microbe. So this is one of the microbes that plays a role in producing short chain fatty acids. So the infected animals have elevated levels of a species that we know can produce short chain fatty acid butyrate, which is something that we expect to see potentially anyway in an inflamed tissue. If we look at gut metabolites between the infected and the controls, and we also do a principal component analysis on the metabolites using essentially metabolomic approaches, we do see a distinct metabolic profile of infected ferrets compared to uninfected ferrets. So there is enough of a difference in the principal components of the metabolites to say that there is a distinct profile in infected animals compared to uninfected animals. And that's exciting to us because now we want to understand what those metabolites are. So we looked at a little bit more detail at those. Several bile salts and short chain fatty acids are increased in the Campy infected ferrets. That's in the red. I want to draw your attention to one of those in particular, butyrate. Butyrate is one of the fatty acids that plays a role in barrier function. It's also stimulated by production or by the growth of Clostridium, and we saw elevated Clostridium. So we're seeing butyrate produced in these infected tissues, and that's going to be something we're going to follow up, but I'm not going to say much more about. David Hendrickson, former postdoc in my lab, who now has his own lab at the University of Texas, has shown that Campylobacter in fact has a butyrate sensor, and so that's an interesting line of investigation that Dave is taking in his group. We also see increased lactate production in these animals, and that's something we're going to look at a lot more over the next few slides. So as I mentioned, in our model, we think that as inflammation occurs, oxygen levels increase, and we also think that there are metabolites that enable Campylobacter to thrive, and one of those that we hypothesize is lactate, and indeed lactate levels are elevated significantly in animals infected with Campylobacter compared to animals just gavaged with PBS. So we're going to explore this lactate. We see no other sort of central metabolites increased in Campylobacter infected animals. In fact, most of them are decreased, and we can talk about why that might be, but we're not going to explore it any further in this talk. We're going to look at this elevation of lactate. So what we think is happening then is in early infection, when oxygen levels are low in a healthy barrier function colonic epithelium, oxygen levels are low because the cells are undergoing oxidative metabolism. There's a little bit of Campylobacter. They're starting to colonize. We don't see much on the epithelial surface, but it's there. As they expand, that's consistent with and coordinated with inflammation in this tissue. We think the inflammation is playing a role, as I showed you in that model slide, of causing disruption in oxidative metabolism, leading to elevated luminal oxygen levels. We've also shown now that lactate levels are elevated, and we think this combination of elevated lactate and oxygen is an environment where Campylobacter can thrive, and that's why we start to see this high level of Campylobacter decorating the surface of the epithelial cell, of the epithelium in infected animals. So let's look a little bit at the lactate metabolism by Campylobacter. Campylobacter has a pathway for lactate uptake and utilization. It has a lactate permease. It has 3 enzymes that play a role in lactate dehydrogenase, driving lactate to pyruvate, and then it goes into the TCA cycle. So lactate can go to pyruvate through this pathway. The genes for that pathway are all encoded on an operon. LCTP is the permease I just told you about. That's the first gene of this operon. These 3 genes play a role as lactate dehydrogenase. So Ratam made a mutation in this first gene. This is an operon. So knocking out this gene actually disrupts the rest of the genes downstream. So knocking out LCTP with a kanamycin insertion leads to a cell that essentially can't grow on lactate. This is the LCTP mutant. Grown in presence of lactate, it essentially doesn't grow. The wild type goes through several doublings in this minimal lactate media just fine. So LCTP is required for lactate-dependent growth. When we look at we ask the second question, which is what about oxygen? Because our model is proposing that it's lactate plus oxygen that enables Campylobacter to thrive in this environment. So if we grow the cells microaerobically, say 10% oxygen instead of 21% oxygen, the wild type and the LCTP mutant, they're both inoculated in these cultures at about 10 to the 7th CFU. Over time, in microaerobic conditions, the wild type grows to several orders of magnitude higher than the inoculum. By 24 hours, it's down a little bit, but still has grown to about 10 to the 10th CFU per mil, whereas the LCTP mutant is diminishing in growth and ultimately is down a couple of orders of magnitude below the wild type. This does not occur if Campylobacter is cultivated anaerobically. The wild type and the LCTP mutant after 24 hours are at the same concentration as they were in the inoculum, and so Campy requires in growth on lactate or mucin plus lactate, in this case we use mucin in the media, requires oxygen in order to replicate. So then Ratam did another very clever experiment where he cultivated the microbes anaerobically for several hours, a day actually, wild type and LCTP mutant in either mucin or mucin plus lactate. They don't grow anaerobically very well at all. If he shifts them to microaerobic conditions now, the wild type cells grow fine, whether they have lactate. They grow a little bit better if they have lactate, but they still grow with mucin alone. But the LCTP mutant does not grow at all. So in this experiment, these cells were just grown individually under these conditions at either microaerobically or anaerobically. In this experiment, Ratam cultivated them under 2 different conditions, first anaerobically, then shifted them to microaerobic. This profile parallels what we see during in vivo expansion, where early on in infection, when we think the lumen is relatively anaerobic because of oxidative metabolism by the host, the colonization levels of Campy in the first 24 hours are really low, after which they begin to climb so that by the end of the experiment, we see they've come through several orders of magnitude of growth when we believe the epithelium is becoming oxygenated because of all the mechanisms that I mentioned earlier in the talk. So this is our working model that the Campy is thriving because of both lactate and the oxygen that's produced during the inflamed growth. So our hypothesis is that lactate and gut luminal oxygen regulate expansion. We've shown that lactate is elevated during this infection. We're hypothesizing oxygen is. We haven't shown that yet, but that's our next set of experiments to prove that oxygen. Actually, we have some markers suggesting that oxygen levels are increased, but we haven't done anything with oxygen electrodes or anything like that. So we began to become interested in understanding how lactate-dependent growth is regulated. So Ratam did an RNA-seq experiment where he looked at all the RNA molecules produced in cells grown either plus or minus lactate and just compared the RNA levels of every gene under these conditions. And what he found was an elevated RNA from a gene that encodes an orphan regulator of a 2-component signaling system. So 2-component signaling systems in bacteria, many of you are aware of those. One component is a sensor molecule that often resides in the membrane, receives a signal. When it receives a signal, it phosphorylates first itself and then a response regulator. The phosphorylated response regulator is often a transcription factor, a DNA binding protein, which upon phosphorylation can become active to bind DNA and either activate or repress transcription. description. What Ratam discovered was a gene we call LCTR, which is an orphan response regulator that has features of a response regulator in a two-component system, but we have not identified any sensor kinase as part of this system. So we're interested in understanding how LCTR works. LCTR, we think, regulates the LCTP promoter region. This is the permease and the downstream dehydrogenase genes. This is the promoter region for that gene, 5' to 3'. 3' on this cartoon ends at the ATG, or it's actually a TTG start site for this for this gene, the LCTP, the first in the operon. We think there are 2 binding sites for LCTR, and I'm going to show you some evidence for that. But based on some work on this orphan regulator, totally unrelated to lactate utilization by another group, we think there are 2 binding sites for LCTR. One is present here. We call this the LCTR box. Another is present there in inverted repeats. And the binding site overlaps the -10 for this promoter where sigma 70 binds. So this would suggest LCTP is acting as a repressor, but because it binds where the activator RNA polymerase would typically act. This is the canonical site for LCTR. Ratam has purified this protein. This is a his tag protein where we put a 6 his tag on, and then we could run the purified or the extracts of this cells expressing this protein over a nickel column, which will bind. We run everything through the nickel column, and everything comes through, and then we can knock off whatever was bound to the nickel column with imidazole, and that's the protein right there. So it's a highly purified protein. We're actually in the process of determining its crystal structure right now, but this is a gel shift experiment with the LCTP promoter. So this is the LCTP promoter that's been labeled. It's run in this gel and it runs to this position. If Ratam mixes this purified protein LCTR with LCTP promoter over different concentrations, increasing concentrations, we see an increasing amount of the LCTP labeled promoter running at this position in the gel, which is a complex with LCTR. If he mixes a different promoter that has nothing to do with lactate utilization in the cell, a gene called MAP-A that we discovered in another study, if he mixes that promoter with LCTR at very high concentrations, it doesn't run in a shifted fashion compared to the free promoter. So LCTR binds specifically to the LCTP promoter. We're testing now, but we predict it binds to these inverted repeats in the LCTP promoter. And so now we're very interested in understanding the role of this protein in regulating lactate-dependent growth in vivo. So to summarize everything, we think that Campylobacter comes into a fairly healthy gut with oxidative metabolism of the host that restricts oxygen in the lumen. And upon induction of inflammation, inflammation causes damage to this tissue, which does 2 things. It changes oxidative metabolism, and it also releases lactate through mechanisms that are complex and we'd like to understand better. And that combination of oxygen and lactate enables Campylobacter to use LCTR regulated LCTP operon to expand to high levels. As I mentioned earlier, but I won't show you the data for, we also have evidence that lactate induces motility of Campylobacter, which enables the bugs to enter the host cells. Motility is an important virulence factor in colonization by Campy. We showed that years ago. So that's the story that we have right now. The questions that are coming up is, does LCTP and lactate utilization play any role in the adherence and invasion of human intestinal epithelial cells per se? We know it's important for thriving in the intestine, but does it play any more role in tissue damage or tissue manipulation by Campylobacter? What is the colonization behavior of the LCTP mutants in ferrets? We haven't tested that yet. Obviously, we need to do that. The LCTP mutants don't grow in the presence of lactate, so we would predict that they don't grow very well in the ferret if our model is correct. What's the source of the elevated lactate in Campylobacter infected ferrets? We'd like to understand that. And finally, what's the mechanism of this regulation by LCTR? Are there other LCTR regulated genes? We have pretty good evidence that those LCTR boxes are found on other genes. We'd like to know what role they play in colonization by Campylobacter and lactate-dependent growth. So that's my story. It's Ratam's story. I think he's done a beautiful job with developing this new model and uncovering all this new physiology for Campylobacter. So thank you for your attention, and I'd be happy to take any questions. And again, I wish we could do this in person, but I really appreciate the invitation to visit your meeting today.
Thank you. Thank you, sir. It was just a brilliant talk. The entire journey of the Campylobacter infection you've discussed. And I think we have a brilliant feedback from all our participants. But I would like to start with a couple of questions. I think Kalia, sir, wants to say something first.
Okay.
So I am curious to know, sir, the lecture was fantastic and the model you have developed is excellent.
Thank you.
Now, two things I want to know. That can we counter the pathogenicity of this Campy by infecting with another microbe, for example, non-pathogenic lactate uptake metabolizing microbe?
Yeah, so that's VC. I think that's a brilliant idea. I mean, so now we understand the physiology. The reason we set up the ferret model was to ask those exact type of questions. If we understand the growth physiology of Campy, not related to its production of toxins or pili or flagella, but just its physiology, can we then do something to manipulate the host so that the physiology isn't available to Campylobacter? And a lactate utilizing microbe is a great idea. I love that idea. I think it's something we're all thinking we'd like to do. Probiotics are, we're all thinking that that's going to be the future of intestinal, controlling intestinal infections. They have not worked really well. They've been tried in poultry because Campylobacter is a human pathogen because it colonizes poultry. So if we can eliminate, out of the billions of chickens that are slaughtered every year, if they didn't have Campylobacter in those chickens, we wouldn't be exposed to it at the levels that we are. So probiotics have been tried in birds to very mixed results. But not, if we can understand at a much more sophisticated level the physiology, then maybe we could try some more clever approaches. So I love your idea, VC. Thank you.
So one more question.
Of course.
Is the behavior of this Campy influenced in the presence of other pathogens, for example, Vibrios or Clostridium?
So that's what we'd like to understand now with the Clostridium. There is some evidence in my colleague Linda Mansfield, who's a veterinarian, had done some studies years ago in pigs, showing that pigs infected with a parasite called Trichurias have an altered, their skewered immune response, and that alters the ability of Campylobacter infections to occur. They become more pro-inflammatory. So these, relationships between other parasites that alter the immune response, innate immune response, and predispose the animal to infection that's been looked at. But whether another microbe, what if we could identify a really nice type 6 producing microbe that kills Campylobacter really well, then we could use it as a weapon against Campylobacter. I like those ideas. It just hasn't
thank you.
Gotten done yet. Phages are obviously another possibility.
Yeah, bacteriophage can be another potential candidate.
Yeah.
So we have another question from a participant.
Yeah, can I ask a question?
Yeah, so I think
Victor, it was a great talk.
Thank you.
But I have a couple of questions.
Okay.
So the gut lumen is quite anaerobic. What is the source of the oxygen? Do you think it leaks from the gut cells, the intestinal cells, epithelial cells?
So there's when the cells are undergoing oxidative metabolism, they're consuming oxygen, and the lumen is not it's not oxygenated. During infection, there's the potential for oxygen production. There's neutrophils, for example, and there could be oxygen from the neutrophils that are called in. There's also a disruption in the basic physiology of the intestine, so that butyrate-dependent oxidative metabolism is disrupted. So oxygen levels begin to rise because it's not being consumed as part of the normal oxidative processes in the host.
I will imagine that it has to come from the blood through the tissue only.
Oh, I'm sorry. It absolutely comes from the blood. Yeah, without question, it's coming from the blood. And so it's in a healthy barrier tissue, it's consumed in the cell, but when that barrier is disrupted, then it can come out.
And why can't they use, why this pathogen cannot use the glucose?
Does not have
They use glucose also along with lactate?
No, they can use fucose, and we have some data on that, but it was already published by Alon Stintsey's group that they can use fucose, but they don't use glucose. They don't have the glycolytic pathway.
But last question. Why the Clostridium increases, although I can understand that butyrate is not being used for energy production, but what is the reason of the Clostridium is?
Why is it increasing?
Increases, isn't it?
Yeah, why it increases is a good question. There's a change in short chain fatty acids and bile acids, and so some of those bile acids probably play a role in clostridial growth as well. But I don't know. Maybe Tom's on the call. Maybe he knows the answer to that.
All right. Thank you. Thank you very
much.
But
I don't know why. I mean, we see the Clostridium come up, but I don't know what the mechanism of that is.
Thank you.
Thank you.
So I have like one last question. Actually, we have quite a lot, but we're really running short of time now.
I understand. I hope I didn't go too far.
So one question, sir. Do we have any data to understand the ecology of Campylobacter interaction with the host microbiota?
Yes, there's a lot of work that's been done on that. And I'll just give you one really interesting experiment. So mice are typically refractory to colonization by Campylobacter. And there's probably a lot of reasons why they are. But one really nice experiment that was done was that after pretty aggressive antibiotic treatment to essentially sterilize the GI tract, the mice were repopulated with a human microbiome. And those mice become susceptible to colonization and inflammation after Campylobacter infection. So there is a connection between the microbiota in humans and the ability to be infected. So where that, I mean, that observation was made. We actually tried to repeat that. We were not able to repeat it, but I believe the results. I think we didn't repeat it because the animals, I think they're different in microbiota to start with. I don't know why we couldn't repeat it, but when we gave the same level of antibiotics to mice to sterilize their GI tract that was done in the PLOS Pathogens paper, we killed the mice. So something else is going on with that experiment, but there is clearly interaction with the microbiome and Campylobacter. That experiment, I think, shows it quite nicely. If you humanize the GI tract microbiome, they become more susceptible to Campylobacter.
Okay, thank you, sir. Thank you so much. You've just captivated the entire audience. Everybody is.
Thank you. You're very kind.
To hear you. I wish we had more time, but I would just formally like to conclude the session by saying big thank you from everybody here. It was great to have you again with us. Thank you so much.
Thank you. Roop, my friend, take care. Nice to see you, sir. You're on mute there, but that's fine. I'll let you get back to your session. Thank you very much, Ankita.
Thank you. Thank you so much.
Bye-bye.
So our next keynote talk is by Dr. Cecilia Alonso from Centro Universitario de la Región Este Roca, Uruguay. Dr. Cecilia is an aquatic microbial ecologist. And before I start with her introduction, I would request Professor Ruplal to welcome her formally.
Thank you, Cecilia, for being with us. We heard you in Hyderabad, and it was a wonderful experience. And you being a aquatic microbiologist, many of us want to hear you again. Thank you very much for being with us and agreeing to our request to participate in this meeting. Thank you.
Thanks to you.
Dr. Cecilia is an aquatic microbial ecologist, associate professor at the Universidad de la República Uruguay. Her research focuses on the ecophysiological changes of bacterial assemblages during environmental transitions to understand how the individual responses of the different bacterial groups ultimately affect functions at the ecosystem level. More recently, her team has developed a line of work for the utilization of the taxonomic and functional structure of bacterial communities as indicators of the environmental health of aquatic ecosystems, focusing on the assessment of emergent contaminants. Today, she will be speaking on bacterial communities as an indicator of environmental status, from proof to principle to environmental management. Over to you, Dr. Alonso.
So good morning, everybody. Good night for me. So I would like first to start acknowledging the organizing committee for inviting me, especially Dr. Ruplal. It was really a pleasure to meet you when I was in India 2 years ago. So I'm really happy. I would be delighted to be there, but I'm very happy and honored to share this work with you. So I will start sharing my Can you see my screen?
Not yet, ma'am. Could you share it? Yeah.
Okay. A second.
Yeah, we can see it, ma'am. Perfect.
Okay. Sorry. So, thank you very much for the nice introduction. And as you mentioned, today I'm going to tell you about an approach that we have learned, in fact, from ecologists that work with macroorganisms, but now, provided the advances in microbial ecology, we can start applying it to microbial communities, and I hope it is of use for some of you. So this is the outline of the talk. I will provide a very brief overview on the use of bacterial indicators of environmental health, and then I will show you a proof of principle of the approach that we are mainly using, that is the indicator value analysis, just to show you how we can classify different estuarine habitats based on their bacterial communities, and then move on to an application that is more devoted to environmental management and particularly for indicating emergent contamination in different aquatic ecosystems. And finally, I will give a brief summary and an invitation to you. So stay tuned. Environmental monitoring is a need from human societies. We need to assess the status of our ecosystems in order to preserve them and in order to take advantage of the functions that we are using from them. So what we need is indicators that are able to reveal the complexity of the environment, to summarize the complexity of the environment, but that they are still rather simple to measure in the large scales that are needed for environmental monitoring, in the large spatial and temporal scales that are needed. So the environmental managers need to use indicators in order to have a cost-effective and a rapid indication of what is going on, something that can also be easily transmitted to the public and to the people that is going to make decisions about the environment. So we all know here that microbial communities are by far the most diverse communities on Earth. And we also know that they easily and rapidly respond to changes in the environment, either altering their taxonomic structure or their functional structure. However, the way in which we assess microbial diversity is rather cumbersome as compared with what people can do with plants or animals. So this, for some time, has impeded that the bacteria are really on the focus to be used as environmental indicators. Luckily, in the last decades, microbial ecology has undergone a major revolution, mainly driven by the rise of the next generation sequences approach. so that we can get a very exhaustive and deep insight into the diversity of these communities, into the taxonomic and the functional diversity of these communities, without the limits that were imposed by the cultivation approaches that were the first approaches that we could use to deal with these organisms. But as you know, worldwide, in the legislation, still the indicators that are used, for instance, for water quality assessments are based on culturable bacteria. You know that like the Escherichia coli, or more general, the coliforms, or the Enterococci. So now that we know so much about the diversity of these communities, I think it's time to fill this gap between what we know from the basic research and what is there on the legislation. So this is what we are trying to do. And this is what I'm going to show you, a little piece of what we are contributing in this field. So how do we find alternative indicators of environmental quality when we are dealing with such complex communities? The first thing that we need to do is go to the environment and gather data, environmental data, that are appropriate for the purpose that we have. We have to measure what are relevant variables to have a habitat categorization. So by using ordination, clustering, partition methods, everything that comes from the ecology approach, from the ecological approach, there is what we gain a lot for what people have been doing in other fields of ecology. So then we can have a categorization of these habitats, and we can know that these habitats are characterized by this or that variables. And then once we have these groups, these different groups of habitats, we can start searching for differential abundance of the different groups in which we are interested, different bacterial groups in this case. I mean, you can do it for anything, but now we are talking about bacteria, we work with bacteria. So for that, you have to run different differential abundance testings. There are many possibilities. One that we like a lot is the indicator value approach that comes again from the macroecologists. It was initially developed for plants. And this index here is what is highlighted here. Has two components, the A and the B component. The A component is maximal when a given species, let's call it species. Now we will then work with OTUs or ACVs, but now let's call it species to make it easier. When a species is only present in a certain habitat, this component is maximal. And then the other component is maximal when this species is present at all the sites that are belonging to these habitats. So to be a perfect indicator, a species needs to be ecologically restricted to a certain habitat and very frequent within it. Once we have selected these candidates to be indicators, then we can move a step further, and is to try to predict if a sample is belonging to this group or to this group or to this group, only based on the bacterial community composition. So we first define those groups based on environmental variables. Then we search for those groups, which bacteria are characteristic for each of them. And then we assay if these bacteria are able to predict to which group a sample belongs without knowing the environmental data, only looking at the microbial communities. So do you think it's possible? Now I show you a proof of principle. We work mainly in coastal areas, in estuaries and in the coastal sea. And the first trial that we did was to check whether we could find indicators of estuarine groups of sites. We worked with our favorite estuary, that is the Rio de la Plata estuary, that is the largest estuary in the world that lies between Argentina and Uruguay. And then we also gather information from the databases, from the publicly available databases, from selecting estuaries that were representative from different continents, in fact. And here you see the physicochemical data that was accompanying the bacterial community composition data that we had for all these estuaries. So based in this physicochemical data, we defined different groups within each one of the estuaries, and then applied this INVAL approach, the indicator value analysis approach, to check whether we could find indicators that could tell us to which group each sample belonged. So these are the results for the Rio de la Plata. For the Rio de la Plata, we could define 4 different groups. These groups were statistically significantly different. So they based on the physicochemical properties. For instance, this group was composed from samples from the middle estuary and from the lower estuary, from the lower end, that is the marine end. These were from the middle estuary. These were samples only from the upper estuary, that is the freshwater end, and these were samples from the lower end of the estuaries, that is the marine end. And what we have here is these are all the samples, and this is to which group they belonged according to the physicochemical data. And these here are the probabilities that each of the indicators of each one of those groups, was given to that sample of belonging to their group. So in this case, for instance, I don't know if you can see it, the true group of this sample assigned by the physicochemical data was number 1, and the probability given by the indicator of group number 1 was 1, that is 100%, and the others say 0. So they say this sample does not belong to my group. So effectively, this sample was assigned to group 1, that is where it belonged. And the same was true for all the samples here. In this case, all samples were correctly classified by the bacterial indicators. Here we run the same analysis for the other 3 estuaries. As you can see, for one of the estuaries, we again got all samples perfectly classified. And in the other 2 cases, some samples could not be perfectly classified, either because 2 indicators had the same prediction, and then we could not assign it to a group, like in this case, or in other cases, because the indicator of the wrong group, let's say, was more powerful in the prediction, and then the sample was misclassified. But in any case, as you can see, most of the samples are perfectly classified. And then we went one step further, and we combined all this data in one single database, and then we assign each estuary as one group. So Rio de la Plata estuary was group 1, group 2, and so on. And then we search for indicators, for bacterial indicators, and check whether they could recognize their own estuary in this mixed database. And as it was a quite large database, we could start applying other methods, machine learning methods, in which you can use make a partition of the data, a random partition of the data, and then use a training set to train the models. Here we use different models, this random forest, linear discriminant analysis, neural network, support vector machines, decision trees. There are many more, we tried those. And then once you train the model, you have a new data set that was not used for training the model, in which you test the prediction. And as you see here, here's how these samples from the dataset were, from the test dataset were classified. Some of them classified perfectly and some of them not. But overall, all the methods had 95% of accuracy. So they could rightly classify 95% of the samples. Depending on which method you use is which samples were perfectly classified or not, but overall they performed really well. So having this, we were very encouraged and decided to go for an application to environmental management. So we had an agreement of work with the National Direction for Environment to search for, I will move this, sorry, to search for alternative indicators of emerging contamination. Emerging contaminants are contaminants that are, chemically, they are very different. They can be pesticides, they can be hormones, they can be pharmaceuticals, they can derive from the industry. But what they have in common is that they are not regulated. Normally, they are not monitored, and we still don't know much about their effect in the environment. But there are watch lists that aim to give a follow of these substances, and depending on what are the findings about them, propose them to be monitored in water quality assessment. So we work, this is the Rio de la Plata estuary, and we work here in this region. That is, in fact, this is where I live. I'm here in this small town right at the Atlantic coast. And here is a protected area, a national protected area. And there is a series of coastal lagoons that periodically connect to the Atlantic Ocean. So we work in the basins of these two lagoons. Here you can see a detailed map with the land use around those lagoons. This one, and this is the other. There is, as I mentioned, it's a protected area, so quite some of the landscape is preserved. Here you have wetlands, extensive wetlands. We also have natural grassland, but there is also agriculture going on, there is afforestation going on, and there is also livestock farming. And here we have two cities, this one and this one. In fact, they are small cities, especially for India. This is really small. The biggest city is this one, and it's not even 30,000 people. They are small, but they have very poor wastewater treatment. So these basins are impacted by the agriculture and also by the cities. So what we did is that we defined 23 sampling sites that were encompassing from the streams, going through the lagoons, and then down to the coastal sea. And we sampled these sites 4 times, one per season during 1 year. And we evaluated the emergent contaminants and the microbial communities for the same samples. So the emergent contaminants were, I don't know where to put this, sorry. The emergent contaminants were evaluated using liquid chromatography coupled with high resolution mass spectrometry. And this allows us to screen for almost 400 emergent contaminants, including different categories that I will show you in a minute. We could identify 56 contaminants fully identified, plus other 26 that were defined as tentative candidates. And among them, we could identify the abuse drugs, antibiotics, hormones, products of lifestyle that are called like personal care products, or caffeine or nicotine, pharmaceuticals, and different pesticides, fungicides, herbicides, insecticides. Having this information on the concentration and identity of the contaminants in these two basins. We ran a risk analysis based on the risk quotient approach. That is that you calculate this quotient like the ratio between the maximal concentration that you find of a given contaminant and the predicted concentration that has no effect. When this is below 1, you consider that there is low or no risk, and when it is above 1, you consider that it could be a potential risk. Here in this map, the gray circles are proportional to the size of the gray circles is proportional to the risk, to this ratio. And as you see, here is one of the cities and here is the other city. Downstream the city is where you find the higher risk, especially in this basin, which, by the way, is the one that has the poorest wastewater treatment. So then what we did, as I mentioned before, based on the type of contaminant and on its concentration, we ran clustering and partition analysis, and then you can see here that the samples clearly split in two groups, two broad groups. This is the group that is highly impacted, the group that is harboring the sites that are just downstream the cities, and these are the rest of the groups. So what we did is that we searched for indicators, bacterial indicators of these two groups. And we did that searching for taxonomic indicators by the standard Illumina sequences of the 16S RNR gene, the B4 region. And then we also searched for functional indicators by performing shotgun sequencing and then identifying and annotating the open reading frames. We did that with custom pipelines that are freely available under GitHub. You are very welcome to go for them if you are interested. And what we find is that we could identify bacterial indicators, taxonomic indicators, that means from the taxonomic composition of the microbial communities, and also functional indicators. Here are the indicators for the group 1, the highly impacted group, the taxonomic indicators, and for the rest, that is from low to medium impact. And this is the same for the functional indicators. As you see here, if you remember the inval, the indicator value, goes between 0 and 1. And what we find here is that these values, particularly these ones, the ones associated with group number 1, are very high in comparison to what we find in the literature for other communities. And as you see here in this highly impacted site, you find some of these suspects that are typically found in the human gut or in wastewater discharges. So it makes sense with the ecology of the known ecology of the groups that they are acting as indicators of highly impacted sites. Whereas in the other, you find more like the common bacteria that are thriving in aquatic environments. So having these indicators, then of course we went for prediction, and here are the graphics showing the prediction of the membership of the different samples to the different groups. And as you see here, this is the prediction of group number 1 by the functional indicators and by the taxonomic indicators, and all samples from group number 1, the highly impacted group, were perfectly assigned by both kind of indicators. For group 2, the functional indicators, again, perfectly assign all samples, and the taxonomic indicators perfectly assign most of the samples, 85% of the samples, whereas still 15% of the samples could not be assigned. we were happy that they were not wrongly assigned, at least. It's just that the indicators could not tell to which group they belong. And then finally, we wanted to compare with known indicators with what is in the legislation. So here we are focusing on the prediction of these highly impacted sites that are these ones. The size of the circle is the degree of impact, based on the risk analysis. And you can see here the stars are showing you whether the bacterial indicators signal these sites as highly impacted. And this is what we got. When you look at the fecal coliforms, that is this is what we have in the legislation. At least I guess probably is the same in India. They could not detect this high degree of contamination by these emerging contaminants, and they were only once out of the norm that it was here, but this is not a highly impacted site from the chemical point of view. And we also compared to benthic invertebrates that are also quite commonly used, but the benthic invertebrates did not find anything wrong in these samples. So to sum up, what I would like you to get from this talk is that now that we have this possibility of using the massive sequence technology, this gives us the opportunity to have a deep knowledge of the diversity, of the taxonomic and functional diversity of the microbes in situ, in the environment. And this opens up the opportunity to use approaches that are coming from ecology, from traditional ecology, that until we did not have this view on the bacterial diversity, we could not use them. But now we can, and they are very robust, and they have been used for decades by now, and they have been very refined, and now they are available to us as well. And then we believe that these taxonomic and functional bacterial indicators are promissory candidates. I mean, we knew it from how they respond to the environmental changes. We all know that. But now when you run analyses that are specifically devoted to find indicators, you see that they perform really well, and they might perform even better. Probably they will perform better than the classical indicators that we are still using. But of course, more research is needed to prove this. And I would like to stress that more research is needed that is done in a multidisciplinary approach. Because as you see here, we had to work with chemists to get this analysis of the contaminants done. We had to have people working with the coliforms. We have to have people working with the invertebrates, and we had to have people that were able to make analysis of land use. And this, of course, one single team with of microbial ecologists or whatever cannot do it. So we really need to expand and work with other disciplines. And finally, something that was really surprising for us is that when we were looking in the databases to gather data from the estuaries for the proof of principle from the first work that we did with that, we were very surprised on how little information is there. Sometimes, I think we all have the feeling that we have sequenced a lot, and indeed we have sequenced a lot. But then when you would like to have this in a database that you can work with, you find that the sequences are coming from different regions of the 16S, and then they are not easily comparable. And that often the data of the microbial community structure is not accompanied by the metadata, the environmental data. And then there are very, very few cases in which you have both, and then you can run this kind of analysis. So we still need, despite we have sequenced a lot, we still need to sequence more, and especially there are some habitats that are certainly underrepresented, and we have to make an effort into loading this metadata along with the sequences, because this is what gives us the solid ground to develop and improve this kind of approaches. So I will take a second to acknowledge my team. This is the group of people that has been working in these studies. And as you know, there are many people. As I already mentioned, they have very different profiles, and they are also from different institutions. And this is something that I also would like to stress, that is, I mean, we all know that, but this, I always like to stress it, how important it is to go for international collaboration, because sometimes, I mean, from national, regional, and international collaboration, because it's very difficult for a single group to have everything that is needed to run this kind of complex studies. And environmental studies are always complex because they have multiple dimensions, and this is only possible to approach when you have strong teams with different profiles and giving different approaches to the topic. And finally, the invitation. In a month from now, it will be held the first international conference on the use of DNA for water quality assessment. This comes from a network that it's called DNA Aqua. Sorry, these are my dogs. It comes from a network which is called DNA Aqua, and its main objective was to gather all the information about protocols and about analysis and all the pipelines that you need to analyze this kind of data, and search for novel indicators of water quality with the aim of taking it to the legislation, to put it in practice, to really change the indicators that we are using to monitor water quality. It's by no means devoted only to microbes. It's much broader than microbes. It's focused on molecular approaches to do environmental monitoring. But I think it's very interesting, and the good news is that we are still on time to register until March 1, and also still on time to send abstracts because it closes next Sunday. So you are very welcome to join. It's fully virtual, and is that you are interested in this, and that we can continue discussing in this also in this symposium. And with this, I leave you. I thank you very much. I'm very glad to take your questions, and Dhanyavad.
Thank you, ma'am. Sir, please go ahead.
So, thank you very much. It was a very nice lecture and very positive with very good results. So you had no negative impact on your analysis. That's very interesting. I have a small query that can we generate markers in soils for specific crops against specific pathogens, plant pathogens?
Sorry, I didn't get it. Could you please repeat it?
Can we get markers in soil for specific crops against specific pathogens, plant pathogens?
Well, I mean, this work is focused on finding the relation of these microbes with different habitats. You can use it for whatever. So if you are, for instance, if you are working with plants and you would like to know which members of the microbial community in the soil are linked to any condition, like to a stress, wherever stress you find in the plants, you can use this approach. This approach has been used, for instance, in the soil microbial ecology to differentiate communities that develop under different management, like organic management or like the traditional management. So you can use it for wherever. It's something that just can tell you which of these enormous list of OTUs that you get are linked to which environmental condition.
Thank you very much.
The clue is to very finely define the condition for which you have an indicator, because you will get indicators for everything. So the important thing is that you are clear in which is the habitat type that you would like to indicate. And then you will find them, I bet.
Thank you.
Thank you, ma'am. Sorry, because of paucity of time, we have to go to our next speaker. I would just like to thank you so much for their brilliant talk, your didactic, enlightening talk, shedding light on the major roles that the minuscule bacterial communities play as bioindicators of the environment. Thank you so much, ma'am, for taking out time and being with us here at the conference today. Thank you.
Thanks to you. I will ask the questions in the chat because I have some questions. So
thank you
thanks
so much
for the invitation.
Thank you. Next, we have Professor Joel Acosta with us in the series of keynote lectures. I request Professor Ruplal to officially welcome him before I introduce him.
Sorry, Professor Joel, for the confusion. In fact, this was a mistake on our part, and thank you very much for being with us. We know your work and great work that you are doing on environmental ecology. And in fact, more work we know about, we are very closely to this work because of the association with Om Prakash, who really was there with you for a couple of years. Now, Professor Joel he's a professor and associate chair at Georgia Institute of Technology. Will not take much time, but then hand over the mic to Ankita to introduce formally Professor Joel.
Thank you, sir. Before I introduce him, just want to announce that room B and C are parallelly online with the AMI-INSCR Innovative Research Session for oral presentations right now. Simultaneously, we are running both the rooms. Professor Joel Acosta is Professor and Associate Chair for Research in the School of Biological Sciences and Earth and Atmospheric Sciences at Georgia Institute of Technology. His research group specializes in characterizing the role of microorganisms in benefiting ecosystems and society, especially in the context of climate change and bioremediation. Professor Koska has published 126 referred journal publications and achieved an H-index of 61 with more than 12,440 citations. Currently, he serves chair of Gordon Research Conference in Applied and Environmental Microbiology. He is a member of the American Academy of Microbiology. As Associate Chair for Research in the School of Biological Sciences, he leads strategic planning for 53 tenure track faculty. Today, he will be speaking on, a movable feast, the role of microbes in response to the Deepwater Horizon oil spill in the Gulf of Mexico. I request Professor Koska to deliver his talk.
Thank you. Can you hear me okay?
So we can't see your video. Video is not there.
Yeah, I know. I tested it earlier and I could see myself, but now for some reason I can't see myself. I don't know. I have the camera on.
Okay. Sir, could you maybe just
Joel, can you put it off and can you put this restart your this thing and video?
Sir, could you log out of Zoom and log in again? I think then the video probably would be visible again.
Or else,
Okay.
Ankita, you have the video, sorry, the PPT already with you.
Yes,
you can
sir.
play it.
Slides.
Okay, so I have my slides ready, so I'll leave and come back real quick here.
Okay, sir.
Is that what you want me to do?
I have your slides. If you want me, I'll just share the screen and play them for you.
Yeah, okay, that's fine.
Okay, I'll just do that, sir.
And let me see if I can share my screen first so I can handle the slides. Hold on.
Please go ahead.
Okay. Okay, can you see the slides?
Yes, perfect.
Yes.
I can see them.
Okay. Yeah, I'm sorry, I don't understand.
We can
Oh,
see
now
them.
you can see me. For some reason it came on. I don't know why.
Yeah. Thank you.
Okay, good. All right. So I'm sorry that you couldn't see me before. But I thank you very much to Professor Lal for inviting me to give this talk. I apologize that I was not there yesterday, and I apologize for the mix-up, and I hope that I did not inconvenience you all. So thank you very much for being here tonight. So I'm going to take you on a journey to discuss the research on the role of microorganisms in the response to the Deepwater Horizon oil spill, this journey that we've been on for the last 10 years since this major oil spill. So together with Mandy Joy and Rita Colwell, I chaired a legacy synthesis group on research that was conducted under the auspices of the Gulf of Mexico Research Initiative. This was a $500 million program to study the ecosystem effects of the spill and the remediation of the spill. And so I just want to point you to this report that was supported by the American Society of Microbiology and the American Academy of Microbiology. We had an international colloquium, and then the report came out last year. So I won't get a chance to talk about all of our research on this spill, so please take a look at this report. The link is at the bottom of the slide there. I also published last year a synthesis of all the microbial genomics research that we've done on this spill over the last 10 years, so please look at that article. And you may know that there was a Hollywood film that was produced on the Deepwater Horizon, but I unfortunately was not in that movie. So, okay, let's go on. Okay, so I want to present a case study of what happens basically when oil comes ashore onto beaches. We had a long-term study of the fate of oil on beaches, and I'll get to that later. First, I just want to thank my collaborators, Marcus Hoodle from Florida State University, Kostas Konstantinidis from here at Georgia Tech. This is very much a team effort that we've all worked on over the years, and I want to thank the Gulf of Mexico Research Initiative for funding this work. So I call this a movable feast, and I do so not only because I'm a big fan of Ernest Hemingway, but also because it's a metaphor for change over time. And as we'll see, hydrocarbon degrading bacteria and those populations in nature change with time according to oil chemistry. So I want to start with this map of the Gulf of Mexico. The Gulf of Mexico is a relatively small ocean basin off the coast of the southern United States. And you can see Florida, for example, on the right-hand side here, the state of Florida, the state of Louisiana in the middle and the north here. I show this slide to show how important oil and gas is to the United States. It's very important to the economy. A large percentage of petroleum comes out of this basin. All of the red dots that you see on this slide are existing oil and gas rigs. And all of the shaded areas are leasing sites for new oil and gas. Okay, so it's very important to the economy. And if you look at the green areas in the top here of the slide, these are all marshes. These are all wetlands that support the fishery. Also, a large percentage of fish and shellfish that we eat in the United States comes out of these very sensitive ecosystems. So we have to balance extracting these important oil resources with protecting the environment. And that's the motivation for this research. I also want to provide you with a few facts on the Deepwater Horizon oil spill. This was the largest release of oil into the ocean in recorded history, and also the largest emergency response effort. It happened in 2010 in April, and what I'm showing here is the oil rig that is on fire and sank in April of 2010. Then oil leaked out from the bottom of the ocean for almost 3 months. A total of 5 million barrels of oil were released into the ocean, and almost 2 million gallons of dispersant were applied. Chemical dispersant was applied to try to break up the oil droplets into smaller droplets so that we could protect sensitive coastal ecosystems that I was talking about. And I would be remiss if I didn't mention that it was a terrible tragedy and that 11 people died in this disaster. So I don't have a lot of time to talk about the fate of the oil, but I wanted to say a few things. This was the result of a broken pipe when the pipe was broken off of the drill rig. So on the bottom you see the broken riser pipe and the blowout preventer here. And what you should realize is that when these oil deposits are present in the subsea floor, they're present at incredible temperature and pressure. So when they are released into the deep ocean, they're kind of sprayed into the deep ocean, and you get a range of different droplet sizes. And in this particular spill in the deep ocean, we got a lot of small droplets which were actually neutrally buoyant, so they didn't rise to the surface. So we had these huge deep plumes that were moving around the deep ocean and contaminating the benthic ecosystems. And then you also have larger droplets that rose to the surface of the ocean and formed oil slicks. Quite a bit of the oil, maybe 15 to 20%, was recovered by these drill ships. Some was recovered by skimming, some was burned, some evaporated. A lot of the oil still remained in the deep ocean, attached to the sediments. Some we couldn't track at all. So I want to show you some pictures next of this disaster. So this is the oil rig on fire in 2010. They're trying to put out the fire. These are the oil slicks in summer of 2010, spread out over large areas of the Gulf of Mexico. And then this is the oil burning that took place at the beginning of the response efforts. When that much oil is present on the surface of the ocean, there's a lot of volatile compounds that are harmful to people that are out there trying to clean it up. And so they burn the oil so that it wouldn't be harmful to the people. But what you might not know is that oil doesn't burn well on water. So they had to add an accelerant, a napalm-like substance, to get the oil to burn. So there was yet more chemicals that were released to try to clean up the disaster. This is some video footage that was taken by submarine. You can see that the broken pipe was present at 1,500 meters water depth, about a mile below the surface of the ocean. One cannot just go down there and fix problems, dive down and fix problems. Everything has to be done by submarines. You can see the incredible amount of oil that's coming out of the broken pipe, and you can also see that a large amount of natural gas was also released. This was in June of 2010. Okay, so just a little bit of background on hydrocarbon degrading bacteria. I think those of you that are involved with biodegradation know all about this, but we've found that these hydrocarbon degrading bacteria that eat oil basically are ubiquitous. They're present pretty much everywhere on the Earth's surface. They're highly diverse and adapted to consuming oil. A lot of the oil which is released from these major oil spills ends up being biodegraded by microorganisms. And what you might not know is that oil can be toxic to certain microbial groups, and so it can really affect ecosystem services provided by microorganisms, such as nutrient cycles. Again, I don't have a lot of time here, but I just want to make sure we're all on the same page that biodegradation is largely a respiration process. It happens much faster in the presence of oxygen, so this is aerobic respiration. Oil is oxidized to carbon dioxide and intermediates. Oxygen is concomitantly respired. So oxygen is a major variable controlling the degradation of oil. And also, just like any other biological process, oil degraders need major nutrients. And oil is very poor in nutrients. And so wherever we see oil contaminated systems, generally they tend to be nutrient poor systems as well, nutrient limited. So nutrients are also major limiting factors. I don't have time to talk about all of this, but I wanted to say too that it's not just oil eating bacteria that affect the degradation of oil. It's the whole microbial food web that's present in these marine ecosystems. It's also grazers, that is microorganisms that graze on bacteria that eat the oil. It's microorganisms that produce biosurfactants that break down the oil into smaller droplets. It's, of course, viruses that kill bacteria. All of these aspects of the food web affect the fate of the oil. Some other things to remember about my talk here is that this was the Deepwater Horizon disaster was the first major oil spill since next generation sequencing technologies were available. So what that meant was that we had a much better idea of who's there, what microorganisms were present in these oil contaminated ecosystems. And also, at the time that we started this work in 2010, there was very little metagenomic data to go on in these systems. So I want to give this case study for oil contaminated beaches in Florida. And so what I'm showing here is the gray area is the extent of the oil slicks in summer of 2010. The red dot is where the broken pipe was, the oil rig. The yellow circle is where we sampled at Pensacola Beach in Florida. And we also had a reference site which was not affected by oil at St. George Island. This is a visual history of what happened when oil came ashore on these beaches. So if you look in the upper left-hand corner here, you see the beach in the summer in Florida is a very hot place. And so when oil came ashore on the beach, it heated up and pooled on the surface of the beach, and then parts of the oil then filtered down into the sands. Okay. And then there were a couple of tropical storms that happened in summer of 2010, and those storms acted to bury those oil layers about a meter down in the sand column. Okay. By October of 2010, the oil layers were still there, and it was decided by BP that they would so-called deep clean the beach. And this was a physical cleaning where they bring in big road machines and they dig up the contaminated sand, they pulverize it, they sieve it, they mix it with clean sand, and then they put it back out onto the beach. Okay, and so what this acted to do was basically just to mix up the contaminated sand over larger areas in the beach. But then by, say, April of 2011, much of that oil was degraded and the communities were back to normal. So here, this shows my students out there sampling. We took beach sand cores to study the oil contamination, and then we also dug these trenches and sampled into the beach face, as you see here. So I want to show you some data now from our studies of these contaminated beach sands. First, I'm showing the environmental conditions. So I said that the beach was a very hot environment. And so on the top panel here, you can see from summer of 2010 to summer of 2011 that the lowest the temperature goes in the beach sand is 10 to 15 degrees Celsius, and air temperatures in the summer reach over 40 degrees Celsius. So it's a very warm system. And we were also able to profile oxygen concentrations in the beach sands, as you see here on the bottom panel, with oxygen optodes. And even in oil sand layers, we saw plenty of oxygen for aerobic respiration. So here I'm showing you hydrocarbon contamination data to show you what happened to the oil chemistry. And what we found, the short answer is that oil was degraded very fast, most of it in the first 4 months after oil came ashore in the beach sands, and that after 1 year, all of the oil was degraded and the microbial communities returned back to normal or back to baseline. So here, if you look in this panel, this is the oil chemistry with depth in the beach sands, aliphatic hydrocarbons, aromatic hydrocarbons, and then total petroleum hydrocarbons all show the same story. The darker colors indicate higher concentrations of oil, and so you see oil contamination in the surface of the beach sand in June and July of 2010. That oil then moves down into the sediment column as it's buried by storms, and then it's rapidly degraded in the first 4 to 6 months by microbial degradation. So it's a good story in this environment that this oil was degraded fairly rapidly. We also saw a huge stimulation in microbial abundance, what we call a bacterial bloom. And so here I'm showing a core that we took from the beach sand. You can see the oil layer clearly about 25 centimeters down in this course. It wasn't a full meter, it was about half, 25 centimeters to 50 centimeters here. And what we see here is a huge increase in microbial abundance in that oil layer, and that over 50% of the microbial community here, profiled by small subunit ribosomal RNA gene sequencing, is comprised of just this one oil-degrading bacterium, in this case, Marinobacter. So these communities really changed in the presence of oil, and they were enriched in hydrocarbon-degrading bacteria. If you look in the bottom here, there's again from summer of 2010 to summer of 2011 with depth in the beach sand. Just as I showed you with the oil chemistry data, we see the bubble size here is proportional to microbial abundance. So you see huge increases in microbial numbers in the surface sand that move down with the oil layers, and then the numbers go down as the oil is degraded. The microbial abundance declines. The red line indicates microbial diversity as Shannon diversity, and as we have seen in other oil-contaminated environments, you see a decrease in diversity while the communities are taken over by these hydrocarbon-degrading bacteria, and then that returns to normal after most of the oil is degraded. So, in addition to these huge increases in abundance of microorganisms, we also saw a succession of microbial communities over time in the beach sands, according to the oil chemistry. So early on in fall of 2010, we saw an increase in first responders like alkanovorax, which use alkanes, and then later on we saw other populations which were utilizing the aromatic hydrocarbons in the beach sands. We also saw that some microbial populations declined in oil-contaminated sediment. So, for example, nitrifying bacteria and archaea declined. And so what we think is happening here is that the heterotrophic bacteria were favored and outcompeted these chemolithoautotrophs that are adapted to the low nutrient environment of the beach. I also wanted to acknowledge Dr. Om Prakash here. So Om was a postdoc in my laboratory at the time of the spill, and he was able to isolate and characterize a large number of pure cultures of hydrocarbon-degrading bacteria from these oiled sands. So I'm just showing some of the cultures here. You can see Om behind this flask of or enrichment culture of aerobic hydrocarbon-degrading bacteria. His work really provided a basis for later work that we did with metagenomic analysis, so I want to thank Om. Okay, so we were lucky to be able to create what I think is the largest metagenomic time series to follow what was happening with the hydrocarbon degrading metabolic pathways and controls. And so I'm just showing a snapshot of this data that, from a paper that we published in 2015, where we were able to plot the abundance of genes for hydrocarbon degradation, for the degradation of alkanes, alkane monooxygenase genes, for example, and then I'm not showing it here, but also genes for aromatic hydrocarbon degradation. And so we found that the increase or the abundance of these genes tracked with the microbial succession that I showed you earlier. So there was good agreement between those two data sets. We also found in all of the data, and I won't be able to show you a lot of data that we've collected, in every case where we found where we studied oil-contaminated ecosystems in the Gulf, we also saw that genes for nutrient acquisition were upregulated, especially genes for nitrogen fixation. And so we've studied that in detail. So next I want to tell you a story about a new species of microorganism that we isolated from this system we call Macondamonas diazotrophica. We started out with these 16S data, so these are ribosomal RNA gene amplicon sequence data, again from this time course from 2010 to 2011. If you look at these black colors, you see that this one OTU in our amplicon data sets was dominant in these microbial communities from oiled sands. And so we wanted to understand what this OTU was, what microorganism was representing this OTU. And so in order to do that, we applied a metagenomic approach as you see here, for assembling and binning the metagenomes from the time series that we had. And from this, we were able to collect a lot of metagenome assembled genomes, and then we were able to match the 16S gene from the amplicon data sets to those of the genomes that we collected from the metagenome data sets. And so what we found was that just like with the amplicons, we could find this single species or genome that comprised about 30% of the microbial community in oiled beach sands. When we looked at this genome, we saw that it was a nitrogen fixer. And so we devised a cultivation medium that was nitrogen-free with hexadecane as a sole carbon source, and we were able to isolate this organism and show that the genome that we collected from the metagenomes matched with this isolated organism. And so we named it Macondomonas for Macondo oil that was spilled during the Deepwater Horizon disaster, and Diazotrophica for the fact that it is capable of nitrogen fixation. And we think that this organism is selected in contaminated coastal ecosystems because it can make its own nitrogen source along with consuming oil. And then the student from this work, Smriti Karthikeyan, she then took the 16S gene from this genome and studied data sets of oil-contaminated environments all over the world. And she discovered that in many cases, in many of these oil-contaminated ecosystems, this organism, Macondomonas, was very abundant, but only in the presence of oil, not in the pristine coastal ecosystems. So we think it's important and can be a good bioindicator for oil contamination. A lot of the data that's been collected over the years on oil-contaminated ecosystems is from taxonomic genes, from 16S data. There's much less metagenome data available, and we also know a lot less about the effects of oil on ecosystem processes. And so in order to address that, we did experiments in mesocosms in the laboratory. And so we did it in these chambers that you see up here in the upper left-hand corner. And so for half of the chambers, we used control. We used control conditions, so we added no oil. And then to the other half of the chambers, we added oil in similar concentrations to what we saw at Pensacola Beach, in this environment that we've been studying and where we had the time series for. And what we found was that the nitrogen cycling processes were drastically affected in the contaminated sands. We saw that nitrification, for example, was basically shut down in sands that were contaminated with oil. And conversely, nitrogen fixation was actually stimulated, as I've been talking about. So we made rate measurements in these contaminated sands in comparison to the controls. We could see that the rates were a lot different. And then we also collected metagenomes and metatranscriptomes from these same mesocosm sands, and we could see that transcript abundance agreed with our rate measurements, that we saw high transcripts for nitrification, for example, in the controls, and very low transcript abundance in the oil treatment, for nitrification, and then the opposite for nitrogen fixation. I wanted to end here talking about this genome repository that we created, because I think it's an important tool that came out of our research over the last 10 years. We were lucky enough to collect a lot of metagenome sequence data and advance the field in that way. And then we finished just last year by creating this curated, comprehensible, and searchable database that's global in scale. And so I hope you'll be aware of this database and use it if you're involved in this type of research. And we've now used this database to extract over 2,000 genomes, metagenome assembled genomes from global metagenome sequence databases, and show that over 95% of those recovered genomes represent uncultured and uncultivated species. And so when we started this work, I think the perception was that a lot of the microorganisms that carry out biodegradation had already been characterized. What we're showing here is that we've only scratched the surface of the microbes that are capable of oil degradation. a lot of what's out there in metagenomes has yet to be characterized. Okay, so that's, let's see. That's the end of my talk. I'll conclude there and just say that we have learned over the last 10 years that hydrocarbon degrading bacteria are ubiquitous. They're part of the rare biosphere, so they're present in low abundance in the absence of oil, and in the presence of oil, they greatly increase in abundance and take over the communities. We have been able to characterize novel species of hydrocarbon degrading bacteria, novel pathways. We've got a much better idea of the community dynamics, and we found that oil was shown to disrupt major ecosystem functions, principally the nitrogen cycle. And we think that these advances in metagenomics can really help us to develop better biological indicators and to inform emergency response efforts. So thank you very much for your attention.
Thank you so much, sir. Thank you for your wonderful talk. As we know, the Deepwater Horizon oil spill was the largest accidental spill in the marine environment. Your team has uncovered that the oil was actually rapidly biodegraded in the beach, and the oil chemistry levels are back to the baseline, and microbial communities in the shoreline segments had returned close to their baseline levels on a year after the spill. That's a very important addition to our present knowledge of bioremediation. Thank you so much. And due to paucity of time, we would quickly move to the next session. I request my colleague, Dr. Vipin Gupta, to introduce our first conference session for the day. Thank you, Dr. Koska.
Thank you.
Thank you, Ankita. So with the remarkable start to the third day of the conference, we would now move forward towards the next theme of the conference. And the theme is Advances in Microbial Systematics. I would take this opportunity to introduce our first speaker, Professor Venkata Ramana. Dr. Venkata Ramana is professor at Department of Plant Sciences, University of Hyderabad. He has 36 years of research experience in the area of microbial discoveries and systematics. He has over 236 publications in reputed international and national journals. He is recipient of several honors and awards like Tata Innovation Fellow of DBT, Fellow of National Academy of Sciences, E.K. Janaki Ammal National Award for Microbial Taxonomy in 2016 from the Ministry of Environment, Forest and Climate Change. I request Professor Venkat Ramana, University of Hyderabad, Dr. Suresh Korpole from MTech Chandigarh, Dr. Om Prakash from NCCS Pune to chair the session, and Dr. Charu Tripathi from CMP College, Allahabad University, and Dr. Nitish K. Mahato from Deen Dayal Upadhyay College, University of Delhi to co-chair. I request Professor Ramana to deliver his talk on a need for an extensive and systematic survey of microbiological wealth of India. Over to you, Dr. Ramana.
Thank you very much, Vipin. I will share my screen first.
It is visible, sir.
It is now visible?
Yes, sir.
Okay, good morning, all of you. And let me first of all thank Professor Rupalal for giving me this opportunity to speak about my work and my experience with regard to the microbiological survey, which we have been doing for the last about 25 to 30 years. Based on my experience, I thought I will share some of the important events and the need for a systematic survey, which we have ignored till now, and why we should be doing it in a systematic way. So basically, I divided my talk into four major areas. The first one is about the bacterial taxonomy. The moment we speak about the core taxonomy, many of you may not be liking to listen to my talk, but I just want to summarize saying that we have very few core taxonomists in the country and globally. When compared to the enormous number of microbiologists very few of these microbiologists have concentrated into the core taxonomy, which constitutes not more than 5 to 10% of the global microbiologists. The situation is more pathetic in India, with very, very few groups who are working on the core taxonomy. While majority of the people who are working with microbiology and identification of the microorganisms, they are non-taxonomist, based on either 16S or phenotypes. They try to identify the bacteria. Hence, I call them simply as non-taxonomist. And we have a large group of people who are working on systematics, who greatly touch upon the sequences and assign to the taxon, and simply call them as operational taxonomic units or candidatus taxonomic units. And this work is mainly useful for ecological work. So we have very, very limited taxonomists. I will not go into the details of the taxonomy, but I would like to share that how we started with characterizing the species and where we are going to go in the coming few years. When we started our work in characterizing, we were depending only on some phenotypic characters. Slowly, the concept of polyphasic taxonomy has come, using the chemotaxonomy, and subsequently multiple genes instead of one single gene, 16S rRNA gene. We went into something like 5-6 genes put together and start identifying the taxon. And today we have a large number of taxonomic descriptions which are made on the genome based. Some part of this also based on proteome based. And slowly we are trying to understand the functional aspects of this genome that is nothing but your metabolome based. While the genome based and the proteome based is highly conserved, the metabolome based is highly dynamic. I will not go into the details of that, but now presently we are using for the description of taxon, the genome, phylogenome, and the metabolome, which slowly we are integrating the information of the genomics, proteomics, and the metabolomics, and probably we can call that as integrated taxonomy. And this will lead into what we call it as the future taxonomy, which will be based on the systems-based taxonomy. So polyphasic taxonomy will be very soon replaced with integrated taxonomy. And we are heading towards virtual taxa, description of virtual taxa. We all accept that a large number of these organisms cannot be cultivated. And now we are greatly depending on the metagenome data and also the genomic data. As a result, the information from the epitaxonomic information, which we greatly depend on the environmental factors, the culture architectural factors, metabolic networking, biochemical information, physiological information, and the chemical information of the cell, all can be put under in silico, and we can have a virtual taxa in the future. This is my vision. Of course, it may or may not happen, but we are slowly leading to a virtual taxa descriptions. I will just introduce the role of metabolic pathways and metabolomics in the description of novel taxa. This is one group of organisms which we had been working are nothing but the spirochetes. Very little we have understood about these spirochetes, particularly from our country, where we have been concentrating only on the disease-causing spirochetes, that is nothing but your Leptospira and Borrelia. Unfortunately, we have a large number of spirochetes which are not yet discovered from this country. So we have made some very interesting observation that one of the major pathways for the quinone biosynthesis is nothing but your terpenoid biosynthesis. This pathway is nothing but your IPP pathway. isopentenyl pyrophosphate pathway, which greatly depends on two different metabolic pathways. One is your HMB pathway or mevalonate pathway, or the other one is your methyl erythritol pyrophosphate pathway. So we have a group of spirochetes which are having solely the mevalonate pathway. We have one more group of organisms in the spirochetes which are having the MEP pathway, but we have a very interesting group of organisms of spirochetes which have both the HMG pathway and the MEP pathway. So pathways can be used. Some of the core pathways can be used in differentiating the taxa. The other ones are the lipidomics. We can distinguish the taxa based on the lipid or the total lipids. Till now, we are greatly depending only on the polar lipids, but we can use the total lipids and differentiate the taxa based on the lipidomes. Similarly, fermentation products. We can differentiate the taxa based on the fermentation products. One thing which I would like to conclude on the metabolite analysis, which we have been doing it, that metagenomics and metabolomics share something which is very common. I will not go into the details of this. I just want to go into the guesstimates, where we guesstimate from the metagenome data that less than 1% of the diversity is culturable. In a similar way, my friends, I tell you that there is only 1% of the chemical diversity is known to us. Huge number of chemical diversity is also not understood by us till now. So culturing the yet to be cultivated members, identifying to be yet to be identified chemicals is the challenge for the people who are working with metabolomics and related to the taxonomy. I will share some of my experience after extensive sampling and surveying in the discovery of novel taxa from India. I have traveled across the entire country from north to south, east to west, in search of finding out new taxa. In this, we observed that several groups of the phylum at the phylum level are not yet studied. One of the interesting groups I discuss is about the spirochetes. Spirochetes, only very little is known globally. India is a rich wealth of spirochetes, and all the brown indicating ones are all the ones which are yet to be discovered. While we have concentrated on the metagenomics only with respect to some of the important hotspots like your Chilka Lagoon as well as Lunar Lake, we have totally ignored a large number of habitats in understanding the diversity of these bacteria. So we have a potentially a large number of phylums which are not yet recorded and discovered from our country. Very, very few phyla have been studied. A handful of phyla are well worked out because of the ease for cultivation. Importantly, these include the members of the Proteobacteria, Bacteroidetes, Firmicutes, and to some extent, Actinobacteria. Archaea is also not well studied from this country. Culturomics is one area which has interested us in the discovery of new species. You enrich and go for the meta-enrichment cultures and then try to get new taxa. This can bring out several new members. The other interesting aspect
Sir, you have been muted. Your mic is. Sir, hold on a second.
Dr. Ramana, sir, you have to unmute your, you have muted your speaker, please. Microphone.
Sir, Dr. Ramana, sir, your mic is mute. Yes, yes, it's working, sir.
Now it is all right.
Yes, sir.
How do I go back? Yeah, where it got muted from here?
Next slide, sir. Yes, yes, from here.
Okay.
Yeah, from here, yes.
Yeah. The other interesting aspect is about the holobiomes or holomicrobiomes and metaculturomes. This is one very interesting topic which has attracted us. Today, we say that no life form is independent and greatly depending on the other life forms. Particularly, this is very much applicable for the eukaryotic systems. So we have some yeast model systems which we are working on, where we say that there are a large community of bacteria which are residing inside the yeast. And these are the yeast which are holding these bacteria for unknown reasons. But majority of these bacteria are not cultivable. So host probably has a hold in regulating these endobacteria. Anyway, large things have to be discovered from this holomicrobiomes and metaculturomes. But what we commonly say that there is no eukaryotic life form which is independent, but it is depending on the other prokaryotic systems. So holomicrobiomes and metaculturomes, I want to culture X bacteria, I need to culture invariably the eukaryotic member. As a result, the eukaryotic is growing, the prokaryote is also growing. So this is one area which is picking up now, and we are looking into these aspects. So there is a need for an extensive survey of microorganisms from India. We know that India has two major biological surveys. One is the Botanical Survey of India, which concentrates on the flora, and they have made a lot of advancements with their own objectives. And we will also have on parallel the Zoological Survey of India, which is also establishing the zoological or fauna, wealth of the fauna of a country. These two organizations have done extensively very well, but unfortunately, there is no survey which is made from a country to that extent globally also in a systematic way and recording the entire wealth of the independent individual countries. So microbiological survey needs an integration of many scientists. It is not just a microbiologist alone can do it. Microbiologist needs to be associated with plant taxonomist, zoo taxonomist, geologist for epilithic, endolithic, and many other environmental things. And we also need a lot of chemistry people to be involved and integrate to have a microbial taxonomist. So microbial taxonomist needs an integration of all the other taxonomists and non-taxonomists. We have Botanical Survey of India, Zoological Survey of India working independently. Geological Survey of India also doing a lot of work on the geological survey. But unfortunately, we do not have any survey made till now with regard to the microorganisms. Some amount of this has been put as a human microbiome. This has led to some discoveries related to the prokaryotes, which are related and in association with the human beings. But this is not sufficient, my friends. Let me just give a summary of the current taxonomic status and descriptions. We have almost 280 phylums which are known till now, which includes 145 candidates phylums. We have 3,800 nearly genuses which are recorded, with 150 genera which are candidates genera, and species roughly around 25,000 species are known, out of which 697 are the candidates members. Now, if I ask you what is the status of these things, how many of these are present in India, there is absolutely no answer from anybody, because we have never worked on recording them and putting them into our records. So the estimated bacterial wealth globally is roughly around 1 million species. I call all the cultivable members as assets, and those from the metagenome or yet to be cultivated are liabilities. So if you see the global assets, it is only 0.59 million species, out of which India has contributed and known only for 0.001 million species. The liability is globally, it is 0.94 million species. In India, we have absolutely no records of our liabilities. How many are there? How many, what are the numbers? Absolutely no information. The wealth